Well, Jerry, thank you for making some time here on Doing Aging in 2019. We are this time. Have you been to one of these before?
I went to one of the first ones that Aubrey de Grey organized back in Cambridge, but that was over a decade ago.
Wow. So you've been in the space for a while.
I've been studying the relationship between aging and cancer for most of my career, actually about 25 years.
Okay, very, very interesting. Why are these two topics related?
Well, aging is something everybody can see and recognize, but they don't really understand it. Why do people age? So I've always been interested in what is the underlying mechanism of aging, or one of the most important components of aging, I should say. And what we've discovered is these things that are in the ends of our chromosomes. Maybe I should start a little more basic. That is to say that in every one of our cells, we have these things called chromosomes. All of our genetic information is encoded into 23 pairs of chromosomes. So we get 23 from mom and 23 from dad, and we have 46 chromosomes in each of our cells. And there are ends of these chromosomes. We have such a large genetic genome, as we call it, that instead of having a circular genome, we actually have chromosomes. And if you have chromosomes, that means they have ends. If you have ends, there has to be something at the ends of those chromosomes. And what's at the ends of those chromosomes are called telomeres. Telomeres are these repetitive DNA sequences that don't really encode for genes or things like eye color or how tall you're going to be or things like that. They're simply caps at the ends of our linear chromosomes.
So it's like a buffer?
It's like a buffer. And the important thing about the telomeres is we have to protect those from being recognized as DNA damage. Our cells are really well equipped to recognize DNA damage and repair that damage or get rid of the cell. And if our telomeres look like DNA damage, which they do, we have to cap them with something. And we cap these things with a series of proteins called sheltering proteins. But in 1972, James Watson, the scientist who discovered the structure of DNA, indicated there was a problem. He called it the end replication problem. And the end replication problem basically states the way DNA replicates every time one cell becomes two cells, and during division, that there's a problem at the ends of our chromosomes. There's no way to replicate all the way out to the end. So what happens is every time a cell divides, you lose a little bit of the ends of your chromosomes called telomeres. And over a lifetime, they shorten quite a bit. So this is probably one of the best markers that we have today, in the sense that it measures not how old you are chronologically, but it measures how many times your cells have replicated. So it's more of a biological biomarker of age. And so the question is, why would it be set up that way? Well, it turns out, it's really quite interesting, that there's an enzyme in our body, which we call telomerase, that's turned on during early human development, that keeps those telomeres long and strong, but it gets turned off before you're born, and it stays off unless you get a disease called cancer, and then it turns back on. So this is where the connection between aging and cancer comes. So the question then is, well, why does it turn off? Well, it turns off because we're large and long-lived, and if you're large and long-lived, if you kept telomerase on, you would be at risk for the early development of cancer.
So other mammals, do they have telomerase activated? Or?
Yes. So let me tell you, that's actually one of the more interesting things. So it sort of comes to my new area of research. It turns out that the gene that regulates telomerase is right by a telomere. And I thought that was really strange. It was only about a decade ago that we really started sequencing the genomes of different organisms and things like that. And so once that information became available, the first thing I noticed was the human telomerase gene was right by a telomere. I mean, really close, about a megabase. I mean, we have 3 billion bases or nucleotides in the human genome. And to put this important gene that close suggested it may be mechanistically important. So the next thing we did is looked at what else had been sequenced. And it turns out whales, elephants, big animals, giraffes, they all have their telomerase gene right by a telomere. And it turns out that small, short-lived animals do not.
Oh.
So the question is, so what's going on? So what we believe, and again, we've got some pretty decent evidence to support it now, is that during development, you have rapid growth. You needed to keep your telomeres from shortening prematurely. And there are diseases of telomerase defects and things like that. So some people are born with extremely short telomeres, and we can talk about that in a minute. But what we believe is happening is this telomerase is on to provide for the rapid growth during early human development. And once you reach a certain point, it shuts off. And we think what shuts it off is a new phenomenon that we call telomere looping. What happens is the telomere reaches a certain length. And it's interesting, most humans have about the same length of telomeres when we're born. There could be a little variation, but not much. Part of that could be genetic. And clearly, if your parents lived a long time, you may start with slightly longer telomeres, et cetera. But what we believe happens is the telomere gets to a certain length and then loops over and silences the telomerase gene. And of course, as we age, the telomere gets progressively shorter.
So you build it first. When you're growing, you're building it up, and it gets to a certain length, and it loops over and it blocks.
That's right. Exactly. You got it exactly right. And then as we age, what happens? Those telomeres, due to the end replication problem, get progressively shorter.
So the loop starts to deteriorate.
Right.
And then for the cancer part, then it starts to activate again.
That's correct.
Okay.
So it's not 100%. I mean, it's not like 100% of people don't get cancer. But once it becomes unlooped, it makes the telomerase gene, let's call it, susceptible to being activated.
Yeah.
So things in science are often more complicated than simple explanation, but in general, that's correct. And so our area of research now that we discovered that's how the telomerase gene, I should say there's a theory of aging. It's not very complicated. It's called antagonistic pleiotropy.
Wow.
And what that means, that's a big word. All right, it's a big word. What that means is things that may provide some benefit early in life may have unforeseen consequences late in life. And so that's a good example, telomerase. We have it early in life, we silence it, but late in life, it has this consequence of increasing your risk for cancer. So that's my sort of basic interest. And of course, if telomerase comes on and you have cancer, we have a whole program in fact, we have a program of trying to find novel ways to inhibit this enzyme, this telomerase.
So I was looking for some of your patents before, we started in your research. And in 2002, you had a patent that was trying to identify cancer in the body based on telomerase, correct?
That's right. Oh, we were the first. We developed the first assay to measure that easily in what we call mammalian or human tissues. Before that, it was very complicated assays. They're not impossible to do, but you need a lot of cells. And so what made our test for measuring telomerase in cancer so widely acceptable was it was an easy assay. A pathologist that had tissue banked in their freezers could easily take them out and say, gee, I wonder if my cancer has telomerase. And it turns out that about 90% of all cancers re-express this enzyme. So it's the major pathway for maintaining telomeres. You have to have a mechanism to maintain those telomeres. If they get too short, then the cells will be recognized as damaged and they'll either senesce or they'll undergo apoptosis. So telomerase, evolved to deal with the end replication problem. No question about that. So the next question is, what's the evidence that telomeres is really this clock and does it cause, human disease? And it turns out that there's a whole variety of assays for measuring telomere length, and some are very easy to do and some are very difficult to do. And the easy one to do, unfortunately, don't tell us what we really need. It gives you what we call average telomere length. But we know what's important is not average telomere length, but the shortest telomeres.
And why is that? So is it for instance the percentage of short telomeres, because that's the percentage of potential cancerous?
Well, it could be cancer, but it also could be aging. It could be senescence. Okay.
Oh, right. Okay.
So the theory that evolved way back in the 1950s by a scientist named Leonard Hayflick was that cells taken from the body and put into a laboratory tissue culture environment progressively divide until they growth arrest, called replicative senescence. Hayflick had no idea what caused that, but over the years, people said, gee, it's progressive telomere shortening. And once telomeres reach a really short length, okay, then that signals, damage response. And most cells don't normal cells don't activate telomeres. To get telomeres to reactivate, you have to have multiple changes to get cancer. So, but if you lose certain genes, plus having short telomeres, you can reactivate telomeres. But the vast majority of normal cells just growth arrest or undergo this phenomena called replicative senescence. And so over the years, people have been trying to say, well, do short telomeres cause everything? Or do telomere shortening cause everything? And there's probably thousands of papers saying short telomeres cause Alzheimer's disease, short telomeres cause infertility, short telomeres cause cardiovascular disease. It goes on and on and on and on. And I'm afraid I'm reasonably skeptical of all those results. And the reason I am is because the methods they use to measure telomeres is not measuring the shortest telomeres, but are just measuring average.
So are you interested in, when you're saying short telomeres, you're interested in the normal distribution of these, and basically you're interested in if it's all biased towards the short end? Would you have a critical level by which you don't want to pass?
That's correct. So it's the latter. Once telomeres get to a certain length, and the question is, do you just need one of your 46 chromosomes to be critically short before you're in trouble, or do you need 2 or 3? And so there's some disagreement in the field about that. It's really not important, but to say once you get really short telomeres on some of your chromosomes, that signals stop growing. And so we now know when cells stop growing, bad things or good things could happen. The way I think about it, in a normal cell, to stop growing is really an anti-cancer protection mechanism. That's our first line of protection. So during life, you've got to have tissue renewal, damage repair, and all this sort of stuff. And so shortening of telomeres is fine. But then when they run out, instead of immediately getting cancer, what happens is the cells stop. But again, if these cells sit around long enough, they can do bad things as well. And some of the things.
So that's senescent cells.
That's senescent cells. And so people have come up with this secretory phenotype. It's called senescence-associated secretory phenotype or pathway. And so it turns out some senescent cells, when they get not able to grow and they've sat around for a while, they can cause problems. They can be actually permissive for a cancer cell to emerge. Okay. And that's a big area.
You mean they could eventually convert into a cancer cell?
That's correct. Or they can make another cell adjacent to them become a cancer cell.
Because they're putting out cytokines.
They're secreting things like cytokines, and these cytokines can attract inflammatory cells. Inflammatory cells can force your cells that should not be dividing to start dividing again, and things like that. And it's a very popular area in the anti-aging field now. And people are using what are called senolytic drugs. A number of companies have started with the idea of let's kill those cells and maybe things will be better. And there's been some evidence both ways. Okay. So I'm still on the fence.
You're not sure if we should kill them?
Yeah, well, I think they're not growing for a good reason. It's unfortunate. It's another one of these examples of antagonistic pleiotropy. Things that might be good don't grow, don't have cancer. You need to grow to have cancer. Could turn around to be bad if we get rid of them. It may be protecting us, or it could be that this secretory phenotype could lead to what we call, chronic inflammation. And one of the hallmarks of aging is, in fact, a low-grade inflammation.
Which steadily increases over.
It can keep increasing. In some people, it's really bad. And the way I
think about this from a human perspective, we're really good at acute problems. In other words, you cut yourself, the wound heals, okay? That's fine. But what we're really bad at as a species is what we call chronic diseases, things like inflammatory bowel disease or arthritis. This chronic inflammation is just keeps deteriorating joints and this, that, and the above. So people need to recognize that this chronic inflammation could be one of the hallmarks of cancer. So you recall I was talking about this telomere looping and this one gene telomerase. But what about other genes that might be close to a telomere? So our latest work has shown that there's a whole group of genes that are also being kept in check. That is to say, the cells don't express these genes, and many of them are inflammatory genes. So we now have, for the first time, a mechanism that can understand why, with increased age, you start getting progressively more and more inflammation. Shortening of telomeres, which was keeping those genes basically only on when they needed to be on. Now these genes are on all the time. And a lot of these are the same genes that people have been studying in senescent cells, which they call the secretory phenotype, cytokines and things like that. So this is kind of interesting. And the reason I like this model is it doesn't require necessarily DNA damage to turn these genes on. So it's a counting mechanism. It basically, the field of aging research, at least at the cellular level, says, what happens is your cells divide, and when you get old, they can't divide as fast. It's just like, again, the wound healing. When you're young, you can mobilize a lot of stem cells. They could go there and repair very quickly. As you get older, it takes longer and longer to repair. You still have stem cells. They just can't mobilize as much. But people believe that's due to cells, basically not dying, but being senescent and just can't divide as much. I never liked that model because I don't think where bodies, when you get old like me, basically are full of senescent cells. I mean, I have a few percent.
We don't have an assay to look at that.
Well, nobody's really done it carefully in humans. They've done it in experimental models, which are mostly artificial, where they activate, lots and lots of senescent cells. There's, genetic tricks that you can do to do that. And under those conditions, the data look pretty reasonable. So there's lots of issues here, okay? The first one is, let's go back to the telomere business, okay? So people want to get their telomeres measured. There are companies that will do it for $89 in the US. Other people will charge several $100s.
So there are many companies? I mean, I only came across two myself.
Yeah, I don't know the names that well. I had advised a company in Madrid called LifeLength, and they have what I call a CLIA certified assay. So it's pretty reliable. But the problem with that assay, just like all of them, is you really can't see the absolute shortest telomeres, okay?
Right. So they're looking at the average of all of them, and is it just a white blood cell?
Yes. So what they do is they take a small amount, like a tablespoon of your blood, you put it in a tube, you send it off to some place, and that place prepares the samples. They're then shipped to Madrid, where the laboratories are for this company, and they have a specific assay. They have a hybridization, a fluorescent probe that will only hybridize to telomeres. It's pretty sensitive, very accurate, very reproducible.
Mm-hmm.
The problem is it depends on probe hybridization kinetics. So if you've got telomeres that are really short, those wouldn't come across as a signal. They would you
won't see them.
You won't see the shortest.
Which are the worst.
Yeah, that's right. So again, it's looking.
But can they tell if 20-30% are missing because there's not enough lighting up?
They could, but they're not doing that right
Okay.
now. What they do is they get a number, and they compare your number against people that are basically your age and older and younger, and they see where you fit on the graph, right?
So they basically just count it all up and average the length.
That's right. And they say, gee, you're a 65-year-old, but you've got the telomeres of a 60-year-old or a 70-year-old, right?
Right.
That where you fall on the chart. The trouble is there's lots of heterogeneity. There are people that are 90 years old that have telomeres like a 60-year-old, and there are people that are 90-year-old that look like they
With that assay.
with that assay.
Right.
So we developed a new assay. It's kind of an interesting assay.
When did you do that?
About a year ago. We published it about a year ago, and we call it Tesla.
I saw that.
You saw that?
Cool.
Okay.
Elon Musk will be happy.
Yeah, well, I was hoping for a free car. I'm more worried about his lawyers suing me.
Right.
But anyway, we called it Tesla for the following. It stands for telomere, TE, and then shortest length assay. So that's where it came from, Tesla. And it just seemed that people like acronyms and stuff for assays, and it seems to have caught on. And this assay will actually measure the shortest telomeres. It measures all the telomeres, but it also gives you, it's a low throughput, but it's very informative.
So you're saying, does it capture all of them?
Yes.
And then it gives you what, a percentage of short ones below a critical length that you've decided, or how do you
we still don't really know what the critical length is. I mean, it's interesting. We can see telomeres so short, you almost have no telomeres on some of these chromosomes.
Can you give a distribution?
Sure. We can have the average, the lower 20%, those that are below 1 kilobase, which is really short. There are no assays except Tesla that can measure your telomeres that are 1 kb or less. Okay. So we think as the field matures a little bit further, that people will start using that.
Is it really expensive right now? Is it novel equipment, or is it?
It's skill. It's mostly molecular skills. You have to run gels and this and that, and
it's a little bit intricate.
Yeah, but fortunately, I had a nice mathematician as a graduate student who developed a really good software program. So once you've run the assay, it can do all the quantitation. I mean, you wind up with, literally, think about it, you got 46 chromosomes, 92 ends. So for every cell, you've got 92 bands, basically. One that says, oh, that telomere end is 4 kb, and this telomere end is 3 kb, and this one's 0.8 kb. So, but he developed an assay, so you just take this image into the computer, and it reads out.
And how are you looking at that right now? Are you just looking at the distribution?
We actually look at it as a picture, and then we have the distribution. So, it's very informative. For example, the example I gave at this meeting here in Berlin, I showed a person's normal blood, and from that same person, they had a benign lesion, a polyp that was a precursor to a colon cancer, and this person also had a colon cancer. And so I had DNA from this person's normal cells, their polyp, and their cancer. And when we looked at it, the normal blood was fine. There was really almost no telomeres below like 2 kb. The polyp had about 30 or 40% below 2 kb, and the cancer had 60% below 2 kb. Nobody would ever pick that up.
That's really interesting.
And that's just one example. I mean, we can look at patients that have what we call telomere spectrum disorders or telomeropathies, and they also have a very large load of very short telomeres. And this is where they get into trouble. Some of these, like kids with dyskeratosis congenita, wind up with, bone marrow failure. They come up with anemia. That's how they present. And then they realize that these kids were born with maybe half or less the telomeres that a normal child their age would have.
So now, like, it'd be interesting to talk about causes. So now you're talking about a genetic cause, I'm guessing, that they just...
Yeah, that's genetic. So in other words, to turn on telomerase is not just turning on this one gene. This gene and the telomeres interact with lots and lots of other proteins and things like that. I already told you there's proteins that protect the ends and stuff like that. So anything that is mutated or genetically altered due to inheritance that affects the ability to maintain normal homeostasis of your telomeres from the start go could lead to an early onset disease. And there's some that don't occur until you're 30. Some occur when you're very young, young children, before puberty, 10, 11-year-old people. And it's very serious because we don't really have good ways to treat these children. So that's one of our hopes going forward, is that we can identify which children have the shortest telomeres, and we can figure out a way to maybe transiently elongate their cells, their telomeres. Then maybe that would reduce their risk of getting their disease or
Right. So in this situation, adding some telomerase or boosting telomerase.
Right. So again, there's good ways and bad ways to do that. So we've decided that scientists like to get a big result quickly, and so they would introduce the gene and overexpress it. But there's a risk that too much telomerase could be bad. It's also a possibility putting a gene in could cause what we call insertional mutagenesis. So you could wind up with bigger problems than your genetic disease.
You don't know where it's going to end up, basically.
That's right. So we do it a couple of ways. One is purifying telomerase, and we can then introduce it into cells very easily.
Right, so you just put in as much as you want.
Right. Or you put in what we call the messenger RNA of telomerase, because it won't integrate. So it just goes inside the cell, and like all RNAs, it will be translated into the telomerase protein.
Right, but again, it's a dose, and once it's used, it's used.
No, it goes away. So
right.
it lasts for 2 or 3 days, and it's gone. Okay, so you don't have any alterations of your genome.
Yeah.
And what's good about that is, again, telomerase preferentially works on the shortest telomeres.
Mm-hmm.
And there's a lot of evidence to support that. Again, people still like to report average telomere length, but I find that biologically that doesn't really tell us very much.
So you haven't got your average telomere length?
You mean my own?
Yeah.
Oh, I had LifeLink do it a number of years ago.
Okay.
I was
Just
fine.
once.
Yeah, I'm not.
Do you know what they're missing at the bottom? Is it like the bottom 20%, or because that's really the issue, isn't it? It's not so much the average, but it's the fact that they're missing the worst part.
That's right. If you're missing the very shortest part, then that could lead to what we call genomic instability. You could get 2 chromosomes that fuse to each other, and once you have that, you have big problems. Okay.
Yeah.
So we're still in the study phase to try to decide how short is too short. But we can see telomeres, below a half a kilobase. And again, we start with 10 to 15 kilobases on each of our telomeres, which is sort of interesting. And over a lifetime, when people look at average, they basically say you go from 10 or 15 down to 5 or 6. Well, again, these are assays that by themselves don't really mean very much. When you look with our Tesla assay, it means a lot, because now you can really see what's going on with the shortest telomere. So that's one thing. And we've shown that if you just put telomerase in transiently, we can almost double the lifespan for 2 weeks of treatment.
So is telomerase specific to each cell, or is it just a generic?
It's generic. We have one gene, and some.
And so can you target it to a specific cell? Because you mentioned that there's different areas of the body, right? We spoke about that. Life length and other guys, they're looking at just the white blood cell. Right. You spoke about, 2 tumors and...
Right. So one has to be careful. I mean, taking a pill that activates telomerase in all your cells, it could be fine or it could be dangerous, okay? I think one needs to be cautious about this, because it doesn't take very many people that have a bad outcome of telomerase throughout their body to create a problem for the whole field. Instead, I would give you one example. There's a disease called idiopathic pulmonary fibrosis. This is a disease that when you're 70 years old and maybe you've smoked or something like that, you could get a change in your lungs, so that instead of having good air exchange cells, you wind up with fibrotic tissue, and that creates all kinds of problems. So this mother comes into our clinic, and I'm in Dallas, UT Southwestern, comes into our genetics clinic, very sick, and we measured her telomeres, and gosh, they were short. And so so what do you tell that mother? Well, not much, right? She's already got the disease. We're not good at treating pathology when you really get to it. We should try to prevent the disease from ever occurring. So fortunately, this mom was very concerned about her children inheriting the same disease that she had. And her daughter, we came in, we tested everybody, and her daughter has the exact same alteration that the mother did, but she was only 26. When we measured her telomeres, they were a little bit shorter, but they weren't critically short like the mom. So what do you do for that young lady? She's not going to get cancer anytime soon, but we know it.
But you're expecting her to decline quickly.
Right. And so when she's 40 or so, just like her mom, she's going to wind up with pulmonary fibrosis.
And you think it's a steady process, or do you think it comes along and again it goes off a cliff?
No, it doesn't go off a cliff. It just is a steady decline, and every cell doesn't lose the telomeres at the same rate. So it's a sort of a population kinetic. Some cells might senesce first and others be later.
Right. And so that's my question, because you were talking, like, so first of all is the decision, what do you test in the body? Are you testing blood for these telomeres, and how
Yes,
does that reflect everything
what-
else in the body?
So we started by looking at blood, but we have also had patients that had lung transplants, because that's the only thing you can do for people with this kind of serious lung disease. And when you look at the lungs, they're also very short, just like the blood. So what's in the blood mostly reflects what's going on in your other tissues in your body. Every tissue doesn't divide at the same rate. Blood and your GI, your gastrointestinal tract, and your skin, they turn over a lot during a lifetime. Whereas things like the lungs only turn over with damage and stuff like that. So, but it doesn't matter. You're always replacing cells in your body. So, going back to this young woman with a genetic disease, what would you do for her? That'd be the question. Is there anything? Well, I would tell her, no, don't smoke. That's for sure. And second, I would say you don't need to be hiking up Mount Everest where oxygen is limited. But for her, I think that the risk of putting telomerase maybe just in her lungs might actually prevent her from ever getting this disease. So one can imagine you could take purified telomerase, put it in an aerosol and let her inhale this so it goes through her whole lungs, but it doesn't get into any other tissues in her body. For her, that would probably be something that could prevent it. We don't know that, but because we know that telomerase preferentially works on the shortest telomeres, we have experimental evidence that just a little bit of elongation of the shortest telomeres.
So it's attracted to the shorter.
Yeah, it's just an enzyme kinetics. It actually has been known for years, and we have a lot of experimental evidence to support that you get more elongation. Telomerase
that's good, nice, because it's defensive then.
Right. It's a processive enzyme. So it's an enzyme that doesn't just add a little bit of telomeres in one cell cycle. It can add multiple. And so we don't understand exactly how processive it is and how many repeats it can do, but clearly the shorter your telomeres, the more processive it is on those telomeres. And if your telomeres are long enough, they may not even attract telomerase to their telomeres. And it turns out, if you look at most cancer patients, their telomeres are really short, but being maintained now by telomerase. I mean, you can think about it this way. When that first cell emerges and becomes a cancer cell, what would happen is you can turn on a lot of telomerase, right? And if you do that, you would get these super long telomeres, but that may not be good either. It's hard to replicate the DNA through telomeres. They form these G4 quadruplex structures, and so it may not be good to have too long a telomeres. And what happens if you don't make enough telomerase? Then your telomeres wouldn't be maintained, right? Cells would go back into senescence or die. So what happens, it's not something I figured out, nature figured it out. Most people who get cancer make just the right amount of telomerase to maintain the shortest telomeres. And that's what you see in 90%.
And then it just survives, basically.
And it just survives. It just keeps growing and growing and growing. And so you keep...
So what you want to do that is, you want to do that for healthy cells.
Well, we could try that. That's right. Especially in a situation like kids with dyskeratosis congenita have bone marrow failure. So you can imagine mobilizing some of their bone marrow stem cells, taking them out of the body in a laboratory environment, elongate the telomeres, check them to make sure they're still fine, and give people back their own blood. People do blood transfusions all the time. We have a technique for cancer therapy now called CAR-T, where we take your specific cells out of your blood and we amplify them in a laboratory and make them so they'll recognize your cancer, and we give you back your own cells. So this is totally, legal and stuff that we can do. And for a kid with dyskeratosis, I think the approval agencies like the FDA in the United States would be sympathetic, because these kids have no choices. So, they just are going to get progressively worse and worse. They give them drugs that don't do very much. They can give them, again, a transfusion of somebody else's blood, but then there's always problems with that. So the best of all world is to manipulate this kid's own cells. There would be no immune rejection, and just manipulate them slightly and then give them back. So there's two examples, but you can probably come up with a lot more examples of where, re-elongating telomeres on a cell by cell or a tissue by tissue specific basis could have big medical implications.
So now we have these genetic panels, right? Whole genome sequencing. Right. Do you think you'll get to a point where it would be useful to do whole body telomere sequencing?
I don't think you need to do the whole body. I think if you
because it could be one specific area, like genetically you've got these.
Well, weaknesses. Usually if it's genetic, it's in all of your tissues. Okay, it's rare that you get a sporadic change late in life. We're talking about diseases that you've inherited.
But it seems like there are specific organs often, as with a lot of the diseases, chronic diseases we get.
And we don't understand that.
Right, okay.
There are people with the exact same alteration or mutation, and some people get, intestinal problems, some people get blood problems, some people get lung problems. That's still a black box in our field. And so people are trying to understand why some organs are more affected than others with the same exact alteration. At least we think they have the same exact alteration.
That's not in terms of telomere length as well, though. Like, so if you did an assay of many, many different types of cells, do you think it would be helpful?
People have done that, and they basically find that the tissues another commonly is what we call a skin punch biopsy. So you can look at skin, or you can look at muscle under the skin compared to blood. And when they do that, they're just about all the same. It doesn't really change enough to worry about. But nobody's done that with our Tesla assay. So we don't know if they're...
Right, so they're missing the shortest part, which could be very informational.
So the question is, would it be useful to know your telomere length? Should you be getting your telomeres tested every year or two, like we get our cholesterol tested and things like that? And I don't really have an answer to that yet, okay? I thought when these companies started having these telomere tests that this was for what I would call cocktail appeal. People would go to their parties and say, have you had your telomeres tested? My telomeres are longer than your telomeres, sort of stuff. But I think there's now an interest, especially in people that, let's say it, that have more wealth and can afford these tests, to follow things like that. And the question is, would it be like a tap on the shoulder? So in other words, if my telomeres were at a certain length this year and next year I came in and my telomeres were surprisingly shorter, then
you can ask, what did I do this year?
Right. It's like what behavior did I start eating poorly? Did I quit exercising? So again, we're not trying to change what we already know is good for you. I can give you
it could give you motivation.
Right. Oh, absolutely.
If I had a crazy party year and my telomeres went down 20%, I'd be like, this year I'm going to take it easy.
Exactly. And I have an example of that. When this company in Madrid first started their company, this is before they were CLIA certified, reporters like yourself were very interested. So they came and got their tests done. They did it for free for these reporters. And I got a call from the CEO of Lifeline saying, there's this reporter I need you to talk to. She's a mom with 2 kids, and her telomeres came out like she was 10 years older than she really was. So what do you tell a person like that? I said, well, let's find out what's going on. Well, first of all, she was a smoker. Second, she had the stress of having deadlines for putting her news article, out quickly. And she had a husband that traveled all the time. And so
loneliness, that's a lot of research on that.
So she had all these obvious factors that we know can cause problems. I mean, people have published papers, again, I don't know how accurate a lot of these are, that says if you smoke, you have shorter telomeres. If you're morbidly obese, you might have shorter telomeres. So for her, I said, well, I think you need to think about some life changes. And one would be to quit smoking, first of all. And second, do you ever do exercise or go do Pilates, do something, right? And I think that settled her down. Now, I'm curious to know if she came back. And I said, and Lifeline offered to retest her again a year or so later. But to be quite honest with you, I don't know if she ever came back or not. So,
most of the time they don't, right? It's interesting.
Well, I mean, she just said, well, people bury their heads in the sand.
People like to avoid it.
Right. They may or may not believe, say, well, it's just another silly test, right?
But I think the more we get, the more accurate we get, and the more clear and confident we get about this stuff, it can motivate people eventually, because it just gives them that clarity. I mean, one of the problems today is journalism on the internet and everywhere else, you see different opinions every day. So people are just like, I can't believe anything. I'm not going to do anything.
Exactly. I mean, look, scientists are the same way. We hear stuff at this meeting and said, really? So you just don't know. But my point is that if we know there are genes that are causing, let's say, inflammation, and it's due to short telomeres, we may not want to necessarily re-elongate their telomeres. But if you know what gene is causing this, there could be treatments for, reducing the inflammation. In fact, some of the biggest selling drugs in the world are for arthritis, things like Humira and Enbrel and stuff like that. And that works by not eliminating inflammation, but toning it down a little bit so you don't have progressive disease. And these things sell, $9-10 billion a year in sales. So this is not without reward for these companies. And so everybody understands. In fact, if there was one common theme at this year's conference, is it's amazing how many people talk about inflammation and aging. They even got a term for it, inflammaging. I don't particularly care for that, but it was surprising to me, which nobody ever looked at before we did, is that genes that people discovered for a variety of other reasons are genes that we found from our sort of genome-wide looking at genes that were close to telomeres. And there's a surprising number of genes that are right by telomeres that people have been studying as aging genes. And now we have a mechanism. We can say those genes are getting turned on because of progressive telomere shortening. So it gives you more of a molecular understanding of how this happens. Most people say, bad stuff happens and it's just random damage or this or that. And I'm not a big believer in that. I like to think that because we are reasonably long-lived, that we needed to have mechanism to protect us long enough to reproduce and nurture our children, but we really don't need these mechanisms late in life. And so that's why these genes are getting turned on. And now we have a clocking mechanism that can actually explain part of that. Yeah, that's why it's interesting.
What do you think of the methylation clock in comparison to the telomere?
So people, I know a little bit about that. And people have been studying changes. This is a non-genetic change in your DNA. These are elements that can regulate gene expression at the level what they call methylation or acetylation and things like that. And we've known for 30 or 40 years that methylation changes with age. So if you look at young people versus old people, they actually have differences. It's still a works in progress. There's at least one presentation at this meeting. The guy believes it's really an accurate clock as well. I think at some point I will compare methylation to our telomeres.
That would be an interesting study.
But we haven't done that yet. I mean, it's one of these things I think that one has to it takes a lot of effort to do these things, checking global DNA methylation. I don't think that the current people look at all the genes. I think they have a panel of genes. It's like anything, a predictive biomarker. They might have a chip or something that they can just look at 300 genes and tell you something about your methylation clock. And I think that's reasonable because obviously if you've done it enough times and done it with enough people, then it starts looking like you've got a story there.
A pattern.
Right. Yeah, exactly. So anyway, that's another thing. The one story I haven't told you though, which I think you'll find interesting and hopefully some of your audience, is we just completed a study with very old people that are what we call centenarians, people over 100 years old. A lot of people don't realize it, but in the past, there was almost no people that lived to it.
A quick question. Is it difficult to find those people for studies?
Actually, you'd be surprised. People are pretty proud of being 100, and so they volunteer.
Oh, cool.
And so in the world today, they estimate there's over 500,000 people that are centenarians. That's a big number. And I think a lot of that's attributed to modern medical new interventions. It started with better sanitation and women didn't die in childbirth and better vaccines and antibiotics. If you start going through all the advances we made, including drugs that lower your cholesterol or improve your blood pressure, laparoscopic surgery, you go through all the wonderful advances that have been made over the last 100 years, you can understand that people are now, instead of dying of a lot of diseases like like heart disease are dying of cancer or aging. Okay, so that's why I study this, because these are the big areas that we haven't quite figured out how to control, but we're doing better on cancer than we are on aging. So what we're trying to do is to say we need to intervene in the aging process before you have pathology. If you wait until somebody's is in nursing homes and things like that, it's probably too late to really reverse anything that's already been seriously damaged. You need to maintain your cells and tissues earlier in life. So we did this study, and we had 21 centenarians from a town in northern Italy. It was really wonderful. The average age was 103. Okay.
That's amazing.
Pretty spectacular.
I should go and live there.
Yeah. I don't know what there is about this area. Maybe no stress,
right.
No deadlines. They eat the Mediterranean diet, whatever. So what was interesting about this is we took young people in their 20s and 30s, and then we took a cohort of 60 to 80, and we compared them against these people that were 100 and older. And there's a lot of assays you can do. We did this assay called RNA sequencing. So you look at all the expressed genes in their blood, okay? When we did that, all the young people were clustered in one area. All the old people were clustered in a new. That's good. We sort of expected there would be differences. That wasn't a problem. So what happened to our centenarians? Our centenarians, some were with the young people and some were with the old people.
Wow.
It was totally a surprise.
So you don't really know what to do with that, I guess.
Well, we did, but fortunately, these individuals were from a community where they had good medical records. The people that collected their blood and gave it to us for this study had all their medical records. So I told my postdoctoral trainee then, go back and get us
correlate.
Let's see if the young people had better health than the people that sort of
right,
were similar to
the good health young people.
And it's exactly what happened. The people that were in there with the 80-year-old groups they had poor cognition. They looked at whether they were in nursing homes or not. Most of them were frail or with canes or walkers. Many of them were on multiple medications, whereas the ones that clustered with the young group were almost all living independently. Their cognition was the same as a 60 or 70-year-old, the most healthy, adults. And they had almost no medications. They never get disease. They don't have cardiovascular disease. They don't have cancer. They never see doctors. Just getting blood out of them is something. But the doctors in Italy, they want to make sure these centenarians are healthy. They're very, the people in France are excited. They had the oldest living human being in the world, Madame Jean Telomere.
Yeah.
And now it's controversy about whether she really did live to 122. I don't really care. But there's a lot of national pride in having a large segment of your population old. So the doctors really do try to look after these people if they're healthy. And I can understand that completely. So now that we did that, we said, well, what else is different? Let's go back to the molecular biology. And guess what? Their telomeres were looking just like a 60-year-old, where the frail or low-performing centenarians were all looking like...
This was with your assay?
This is my TESLA.
Right. Wow.
So I went back because we had 21. That wasn't a big thing. I mean, we want to have a bigger population to follow up on this, but it was very interesting. Every single centenarian that was in the high performing group or like young people had very good telomere length. And whereas the people that were frail and in nursing homes, they were just lucky. So it turns out most people that make it to 100 are just plain lucky. They just seem to avoid some of the things that do most of us in, right? They've just never had diseases. And so they just managed to get there, But some of these people...
But you think it's random, or it sounds, if there's a cluster or something...
Yeah, I think that there's probably a genetic component to this. That's something about these people. They had better DNA repair mechanisms, or they inherited something. I mean, we've seen this in certain animals that live a long time. If you think of the bowhead whale that can live over 200 years, they've sequenced the bowhead whale. It's got the telomerase gene by telomere too, but their telomeres are no longer than humans. That's what's sort of interesting. So how come they don't get cancer? They're 1,000 times bigger than a human.
And they never get cancer.
Well, we don't think so.
Right.
It's a big animal, but
yeah.
they if they saw big tumors in these whales, people would know about it, but they don't. And it's the same thing with elephants. They rarely get cancers and stuff like that. So what they've done in elephants and in whales is they've looked, and in the whales they've amplified genes that enhance DNA repair. And in the elephant, they've reported that they have extra tumor suppressor genes like P53. So when you really get down to it, that certain animals in the wild have evolved mechanisms to make them so they can live long and not get cancer. Trouble with humans is very simple. We didn't do that. We don't live today like we lived 200 years or 300 years ago. I mean, even in the past, there was always some people that lived a long time, but we were basically hunters and gatherers and stuff like that. Today, we sit in front of our computers and our TVs and our desks and whatever, and we don't get out and walk 20 miles a day like our ancestors probably did. We've got a lot of things that are going for us, and we don't need to do that necessarily, but we need to recognize we haven't evolved these extra protection mechanisms. That doesn't mean in the future we won't, okay? Just saying these things occurred over hundreds of thousands, if not millions of years, okay? So what we need to do now is to ask, what is it that we could do to make people repair their DNA better so that we don't use up our telomeres too fast? What can we do to prevent the first occurrence of cancer, like some of these other large, long-lived species have done? So I think that's interesting, and I think the studies on healthy aging and telomere biology is interesting. The question you could ask yourself is, these frail centenarians, what can we do for them? And probably not much. But what if you were 60 years old and we measured your telomeres and we said, gee, you look like you're going to be a typical 80-year-old. When you're young and you have less chances of some other disease occurring, that's maybe when we can best intervene. And I think that's something people have been talking about: rejuvenation medicine, cell rejuvenation, tissue rejuvenation. These are really exciting areas. We just haven't gotten sophisticated enough to be able to implement that on a global scale. And fortunately, there are very enlightened people that are very wealthy that recognize this, that it's not about treating late age pathology, but preventing that pathology from ever occurring in the first place. So I think if we can get the resources we need, I'll give you an example, one sort of last example. The National Institutes of Health in the United States funds most of the research, and there's one institute called the National Institutes of Aging, right? And by Congress, this is the old congressmen that are worried about their own health, mandate that the National Institutes of Aging spend most of their money on Alzheimer's disease, end-stage pathology, and basic science of aging. They're only funding about 7% of grants. 7%. You realize how many talented scientists are out there in the world?
So they're funding the pathology.
That's exactly right. So I think it's a poor investment. I'm not saying Alzheimer's isn't important. It certainly is. But again, to me, the key would be to identify somebody at 50 who's likely to get Alzheimer's and do something at that age instead of waiting until they're 60 or 70 when they already have the pathology that may be difficult to reverse. So part of this is my pitch for wealthy people that have the resources and the inspiration to do something about this global problem, to step up to the plate. And fortunately, even at this meeting, there's a number of such people. And so I'm looking forward to continuing my conversations with some of these people, not necessarily for my own.
How can they benefit? They can help this area by funding research?
Well, part of it would be funding research, but probably at a bigger level. So, for example, let me just say, with my studies with these centenarians, what I'd like to do is to get 1,000 people in a trial when they're all 80 years old and track them for 10 years, follow their telomeres every year, or follow these inflammatory genes, so we can intervene in an older.
Would you intervene would it be a randomized controlled trial?
Well, we have to first of all identify if we can predict the year before somebody becomes frail. In other words, if we say, gee, you've got all the biomarkers now that suggest something bad's going to happen in this next year, we have to, first of all, prove that before we
So
start.
just wait and see.
Right.
Unfortunately for them.
Well, maybe
So you can't
not.
tell them the results.
But that's the way clinical trials are done. If you tell people, I mean, there's been enough examples of trials where you think you know what's going on and people get fooled. I mean, they've done what they call double placebo trials. Both arms got the same sugar pill, basically. And when they deconvolved that, they told one set of nurses the pink pill is the good pill, and another set of nurses the blue pill is the good pill. It's, you're going to feel better, you're going to be stronger, and all that. And the people are so easily persuaded. Nobody wants to take the placebo pill in a trial, right? Everybody wants the active ingredient. You don't know if you've got it or not, but if somebody hints at you that maybe you've got the good pill, so hopefully you're being better. We're going to test this and test that. And people
might emotionally be communicating. If the nurse knows which one is which.
I think people's mindset about their own health is important. I think if, there are people that are upbeat, people that are always sort of on the downside, that glass half full, half empty sort of concept. And to me, I think that being positive is important, and it could have an influence. Who knows? There are studies that say women that are raising children that have dreaded diseases have shorter telomeres in their blood. And I think there's some truth to that. Is it reversible? Probably, but nobody knows. They haven't even done that test. Yeah. So it's one thing to get this initial study, but then you need to go there and show cause and effect. And it's very rare in science where people can design experiments to show that. Most people just do what we call correlative studies. Short telomeres correlate with aging. The only way to prove that short telomeres cause aging is to re-elongate the telomeres. And in a laboratory, we've proven that. We put telomeres back into cells, and now they grow forever. They're not cancer cells. They're just like stem cells. And so we know that's correct, but that's a cause and effect relationship now. We need to do that with our clinical trials. We need to say, yes, you're likely to get something really bad happening to you in the next year or two. Mm-hmm. And now we're going to go in there and put you on some anti-inflammatory, or we're going to do this, or we're going to do that. Yeah. And maybe we will prevent that. And we'll just have to see. Unfortunately, with human beings, these are long-term, expensive experiments, but these are the experiments we need to do if we're ever going to make sense of this. Again, many years ago, they did a study on primates on what we call caloric restriction. And hundreds of millions of dollars were spent on this primate, some that got half the calories as the others. One could eat all they wanted, and one were reasonably caloric restricted. And the studies came out mixed. It turned out after all these experiments, they just couldn't say if being on a caloric restricted diet helped that much. I'm not saying it wouldn't. I think there's good evidence that some caloric restriction is good, not only if you're maintaining a healthy body weight, but it may be important for lots of other reasons too. But it's hard to prove that. That's the point. These are still sort of correlative studies. So we need a lifetime of that, controlled studies. We need to get 5,000 human beings that will agree to be on a restricted diet for 10 years versus another group saying, go out and have fun.
So this is going to take a long time.
Oh, for sure.
So one of the things I start to think about is, as our analytical methods get a lot more advanced, are we going to be able to shorten potentially these studies?
It's possible.
If we had a comprehensive view of the body and we actually saw everything.
Right. So that's the reason why I think it's bigger than any one individual scientist. I think we need to get a team of the best, let's call it aging researchers that have a different perspective.
Right.
Somebody interested in senescence and SASP, and somebody in senolytics, and somebody about methylation, and somebody with telomeres, and get around a table and say, if you had unlimited resources, what would you do? And then try to get the smartest people together and to start basically designing what those critical experiments would be. And I think we've got the ability to do that now, but it's going to take
a little organization.
Yeah, I think that's important.
Are you meeting interesting people here?
Oh yeah. There's there are a lot of entrepreneurs that Unfortunately, for most people, it's about how quickly can you start a company and make money and go public and get rich and all that sort of stuff. And I have nothing wrong with that, but it's not my goal. My goal is to come up with better methods and better mechanisms that lead to an increase in the pathology of aging, and if we can intervene in those. But that's not everybody. So you have to sort of sift through motivations of people somewhat. So that's a bit more challenging. But I think the future is bright. I don't want to leave on a negative note. I want to just say that I think the future is very bright for aging research. It used to be people didn't think that aging was something that should be studied. They just said it's inevitable. Everybody's going to age. We shouldn't waste our money on it. But we spend billions and billions of dollars and euros every year on treating pathology that are directly the result of aging.
And it costs us way more, and everyone knows this, but, I think we're a very short-sighted species, unfortunately, most of the time.
Well, anyway, that's about all I have to say for today, but I wish you luck, and your audience, if they have questions, I'm easy to reach. Just Google me. My name is Jerry Shay, S-H-A-Y. You won't have any trouble finding my webpage and things like that if you want to see some of the stuff about telomeres, some basic 101 telomere.
Where's the best stuff to start learning about this? Just
in my homepage, we've got links to things, the literature, a lot of the things that we publish, like our Centenarian study, it's open access. So you just have to go to this journal, Aging Cell, and type in my name.
And all those links should be on your page.
Well, some of them are. If you have open access today, it used to be journals were just they're making money. So now they charge up front and they make it open to the whole world. And I think that's good. I think because you never know who's going to read a paper that I wrote and come up with a brilliant idea and tell me, why don't you do this, that, or the above? And I think that, the group of us, the humanity as a whole, is going to always come up with better solutions than any one individual. So I'm happy to communicate with people. I travel a bit, but not that much. And I'm usually in email touch. So you can find me. I'm not difficult. I don't hide in the corners like some people in their ivory towers do. So again, I look forward to interacting with some of your listening audience. So have a good day. Okay.
Excellent. Thank you, Jerry, for your time.
You bet.